Now, contemporaries following Morgan and Griffith, Avery McLeod McCarthy, Hershey Chase, there was still research going on looking at the structures and looking and trying to figure out what the chromosomes really were. Okay? As I've said in previous lectures for this chapter, they were able to isolate the DNA. They still didn't know exactly what it was, how it looked, what its structures were, but they were working towards it. They knew roughly what the structure of nucleotides were. Okay? They knew this sugar with a nitrogenous rings and a phosphate group. So it wasn't just that Griffith, Avery McLeod McCarthy, and Hershey Chase were working in a vacuum. There were other labs were also working, also studying, but they weren't studying how the information was passed. They were trying to examine how the information was stored. One of the labs happened to be Erwin Chargaff. Okay, 1950, Chargaff was able to publish his findings when he examined DNA. Okay, keep in mind, this is pre-Watson-Crick, so we still don't know exactly the structure of DNA, the double helix that we now know and all that. We just know the components. And one of the things Chargaff was able to figure out, what we now classify as Chargaff's rule, is that when he studied the composition of DNA, he was able to determine that there are some set ratios. Okay? The set ratios were such that for every A, there is a T. That ratio is maintained, and the ratio of G and C's. For every adenine, there's going to be an equal component. For whatever the concentration of adenine, there's going to be an equal concentration of thymine. Whatever the cytosine concentration, there's going to be an equal concentration of guanine. For every A, there is a T. For every G, there is a C. Chargaff's rule. So this is one of those steps, one of those links in the chain that's working our way towards Watson and Crick.